Hi, my name is Yvonne Regel and I'm working at the Department of Medicine 2 at the Ludwig Maximilian University in Munich, Germany.
My research activities focus on epigenetic alterations in pancreatic cancer, and I am particularly interested in the regulation of histone modifications. So epigenetic patterns control the gene expression program of cells and have therefore a great influence on the cellular phenotype. Together with my colleague Dr. Simone Benitz and the contribution of our co-authors, we have described the importance of ring 1b dependent epigenetic remodeling processes in pancreatic cancer formation, and I'm very happy to present our recent publication in Gut as a video abstract. Mouse model studies have shown that pancreatic acinar cells are susceptible towards oncogenic KRAS-mediated transformation, at least under specific conditions. The expression of mutant KRAS in embryonic pancreas cells leads to a spontaneous development of acinar-to-ductal metaplasia and pancreatic cancer. In contrast, the induced expression of mutant KRAS in fully differentiated adult acinar cells reveals no phenotypical changes. Here, an additional pancreatitis-triggered ADM formation is necessary to induce pancreatic carcinogenesis. An interesting aspect is that the ADM cells shut down the expression of acinar sulfate genes and reactivate progenitor genes. We have speculated that epigenetic mechanisms play a role in changing the gene expression profile and therefore promote acinar cell reprogramming and cancer formation. In our study, we set up an in vitro carcinogenesis sequence model containing embryonic and adult acinar cells, as well as ADM and tumor cells. We hereby obtained pure cell populations to monitor the epigenetic changes in each cellular condition. We concentrated our epigenetic analysis on 3 histone modifications. The trimethylation of histone H3 on lysine 4 is associated with an open chromatin and transcriptional active genes. In contrast, the trimethylation of histone H3 on lysine 27 and the monoubiquitination of lysine 119 on histone H2A represents a closed chromatin conformation and a repressed gene transcription. We performed chromatin immunoprecipitation and whole genome sequencing to determine the epigenetic pattern of each cell state. And we additionally assessed transcriptomic changes through microarray analysis. In the next step, we combined the transcriptomic and epigenomic data sets in a comprehensive bioinformatic analysis, and we identified various gene clusters which show dramatic changes in the epigenetic landscape of acinar and tumor-related genes. We described the clusters in our manuscript, and here I will just highlight the most important results. The gene expression profile revealed an increased expression of the epigenetic remodeler enhancer of CEST and ring 1b in embryonic acinar cells, ADM, and tumor cells. Both proteins are part of the polycomb repressor complexes 1 and 2 and catalyze the repressive histone modifications. Interestingly, acinar sulfate genes such as REPJL and KLF15 show an enrichment of the positive histone mark in acinar cells. However, in embryonic acinar cells and ADM cells, the acinar differentiation genes are additionally marked by repressive histone modifications, which is associated with the loss of gene expression. In the following process, the acinar sulfate genes are completely silenced and exhibit only repressive histone marks in the tumor cells. To assess the functional relevance of ring 1b catalyzed gene repression in pancreatic cancer formation, we generated an inducible ring 1b knockout mouse, which is expressing oncogenic KRAS. After inflammation-triggered acinar cell reprogramming, the control mice showed, as expected, the development of ADMs and precursor lesions. However, the ring 1b knockout mice were unaffected, and the acinar cells maintained in a differentiated phenotype, although oncogenic KRAS was present. The epigenetic analysis demonstrated that acinar cells of ring 1b knockout mice, here shown in green, did not enrich any repressive histone modifications on the acinar sulfate genes. In addition, the gene expression level of the acinar differentiation genes remained high in ring 1b knockout mice. So we can conclude that an actively maintained differentiation phenotype is a strong tumor suppressive mechanism and protects the cells of oncogenic KRAS-mediated transformation. In our study, we provided clear evidence that the epigenetic silencing of differentiation genes is a mandatory event in pancreatic carcinogenesis. I thank you very much for listening, and I hope you will enjoy reading our paper.